I really appreciate the introduction from Professor Lin. So it's a great pleasure and honor to be here today, so we can share some of the knowledge and recent progress about the stem cells and exosomes. So particularly, for their potential applications in the skin regeneration and rejuvenation. Okay, so I'm Wenbin Tan, so I'm a tenured faculty. I was in Beckman Laser Institution, and we also have very great collaborations with BRI. Right now, I moved to the South Carolina, University of South Carolina School of Medicine. Okay. So today, so the talk, what I'm going to focus on, so I'm going to talk a little bit different. Okay, so maybe have a different right today. So the first part, I'm going to talk about stem cell biology, and the second part, I'm going to talk about the exosomes and the extracellular vesicles, and the third part, I'm going to talk a little bit about the regulatory compliance. Okay, so what's stem cells? Stem cells is quite, this is the developmental stage of the human embryonic stem cells as the development stage. So at the beginning, so those are the first 8 cell stage. So those are the cells that had the totipotency. And then next, when they move to the 16 cell stage, and they're going to be pluripotency stage. Okay, so and then if we look at the stem cell classifications, and then the first one is going to be the stem cell that they can develop the entire human body or entire human organism. And then the second stage are the embryonic stem cells. They can basically have the pluripotency, and then they can differentiate into all types of the tissues. And later on, and then they're going to have continue to have more specific stem cells, like the neuronal stem cells, and also like the mesenchymal stem cells, etc. The potencies of the stem cell, they have the highest one from the totipotent and then pluripotent. Okay, and then later on you can feel this the track and go to the differentiation process. And then so everybody knows that at the beginning when scientists want to use the human embryonic stem cells to do the research, they face a very serious ethical issues. And then scientists tried to figure out whether we can use somatic cells that can reprogram those somatic differentiate cells back to the stem cells. This, really what happened to this great scientist, okay, so a Japanese scientist, Yamanaka. So he basically discovered an approach that they can redifferentiate the somatic cells back to stem cells. And then in 2005 and 6, and in 2012, and he won the Nobel Prize. So this is a kind of category. I mean, this is a speech what he said in his Nobel Prize, a talk. He said, I was anxious about whether they were really the pluripotent stem cells that we were looking for, because the method used to generate such iPSC cells was much simpler than I had expected. And then in addition, after hearing about a big scandal like that when it happened in a Korean scientist, and then he was very cautious. That's why he postponed his paper for publication for 1 year until he really replicated his results in the human fibroblasts. So, and then published paper back to back on Cell. And then 7 years later, he got the Nobel Prize. So this is a really fundamental a stage in the stem cell biology, because if you can reprogramming the fibroblasts back to stem cells, and then these stem cells basically can differentiate to all types of cells, including like neurons, the endothelial cells, cardiomyocytes, and etc. etc. Okay, so basically the iPSC cells right now is the center, and then they can be used for the basic research model. They can be used for the disease model. They can be used for the drug screening. They can also have a great potentials for the applications in the regenerative medicine. Okay, so Okay, so let's go for this. So this is the current clinical trials using the stem cells. And we can tell that, okay, this blue curve, this is what the recent cases in the clinical trials using the embryonic stem cells. And then recently, I think using the iPSC cells has a spike. Okay, because the iPSC cells can face much, much less of the ethical issues. Okay, so if we look at the skin, disorders, the mesenchymal stem cells is particularly interesting because the mesenchymal stem cells is a type of cell that's going to really generate all types of the cells related to the skin, including the immune cells in the skin as well. So that's why it has great potential for the applications to treat the autoimmune disease in the skin and also the allergic disease involving the skin. So this is the why, because the mesenchymal stem cells you can get from the fat tissue, you can get from the bone marrow, and you can differentiate from the iPSC cells, etc. And then those cells, they can secrete many of the growth factors, like the FGF, VEGF, like the EGF, etc. And those growth factors can promote or facilitate the cell or the skin regeneration and the rejuvenation. So this is a kind of summary of the recent study to see how those mesenchymal stem cells are used in the skin wound healing. But all those are the animal models. I believe that the clinical trials is going to be happening in the next couple of years. So this is what happened. So this is what we did, some research in our laboratory. Then we are basically interested in this Paul-Weinstein and the Sturgeon syndrome. And then everybody, I think, know about this. The Paul-Weinstein and the Sturgeon syndrome, they are congenital vascular malformations. And then we basically got the skin from the patient, and then we generate the iPSC cells. And then those iPSC cells have stem cell marker expression. Okay, all those like the NANAC, okay, so like the 1216, OCT4, and SOX2, etc. And then next, we basically differentiate those iPSC cells to the mesenchymal stem cells, then back to the endothelial cells, because this is what we want. Okay, to look at the blood vessel for normality. Okay, and then those endothelial cells express the stem cell, like the endothelial cell marker, like CD31, CD144. And then when we put those patient endothelial cells on the gel, and the patient actually, those endothelial cells form a much larger blood vessel-like structure and a thicker blood vessel wall than the controls. So this means the patient-derived endothelial cells and iPSC cells, basically they preserved the pathological features from the patients. Okay, then let's move on the next part. Okay, so about the exosomes. Okay. And the exosomes is another hot topic, okay, in the field. So what is the exosomes? So those are the two great scientists, okay, they discovered the exosomes in 1983. So Dr. Johnston and Dr. Holdings. Okay, Dr. Johnston, she was from the McGill University in Canada, and then he was from the Case Western Reserve University. Okay, unfortunately, Dr. Johnston right now, she passed away a couple years ago. However, the discovery made by her students, PhD student Dr. Pan right now, I think Dr. Pan right now is a faculty in the Hong Kong University. I believe Dr. Pan and Dr. Harding is going to win Nobel Prize in the next couple years, or probably, I don't know, probably 10 years. So in 1983, they both published a paper to discover the exosomes that from the reticulocytes. Reticulocytes is a precursor of the red blood cells. Okay, so this is what happens to the what they found. So I think this paper is from the Dr. Holdings. Okay, so they basically found those multivesicle endosomes. They have many of those small vesicles inside, and those small vesicles basically can labeled by the transferrin protein. Okay, and then got released. Okay, so this is how the exosome got discovered. However, during this time, people think, okay, exosome's function is only for the waste disposal, because when the reticulocytes try to mature to be red blood cells, they need to really get rid of all those transferrins, and then looks like the transferrins were disposed by the exosomes. So this is why, since the axon discovered, during the next decade, basically nothing happening until like 13 years later, like in 1996. So this is a very, breakthrough paper published. They show that actually the B lymphocytes, they also secrete axon zones. And then those axon zones have antigens on it. That's basically they have the immunomodulatory, function. So, and this is basically, open another door and then really boost the entire field to move forward. Okay, so let's move on for the axon zones. Axon zones actually is more complicated than what we could expect. So this is a cell, the size is 10 micron, and then they have the axon zone get released from the multivesicle endosomes. At the same time, this cell itself can body out many of other vesicles, like the microvesicles. Okay, so the contents of the vesicles or microvesicles, they have the lipids on the surface. They have the receptors and the spanning proteins. Okay, the inside you may have the microRNA, mRNA, and DNA. So for the human blood, we have usually have 1.5 billion of the axon zones per mil of the of the quantity. So it's pretty high. And then, so I think this is simple, figure, but it's very important because you need to know what happened to the axon zones. A cell, they have 3 types of vesicles. One is the axon zones, the size is 50 to 150 nanometer, and another one is axon zone, so the size is about 100 to nanometer to 1 micron. So those ones are different from these ones, a little bit bigger. So the third one is the apoptotic bodies. So the size is about 50 nanometer to 5 micron. Okay. During your preparation, you basically got all of them. You are not able to really distinguish axon zone from the other 2 types of the vesicles. And then if you look at the contents, and then they are kind of different. Okay, so that's why when you got a product, you say, oh, this is axon zone. No, this is not axon zone. This is actually extra or mixture of everything. Okay, you need to have this concept. So this is another, our study. We just to show. Okay, so in the pore stem blood vessel, they basically secrete many of those, vesicles. The size distribution is about 50 to 500 nanometer. So it's basically have all those 3 types of the vesicles contained. Why? This is another one. You look at the table. It sounds like everything's inside. It's right. It's true because the axon zone basically contain everything inside, most of the proteins. So that's why when you got axon zone products, you think it's a pure product. No, it's not. It's actually, it's a mixture of all kinds of proteins inside. The functions, at the beginning, we think the axon zone is like a trash disposal. Actually, right now, we found axon zone actually is a good, cargo delivery system. It's also a signal convey system among the cells in distance. The function of the axon zones is actually versatile. They have lots of functions. Whatever you can think, it has it. So involving the physiological and also pathological processes as well. And then we go back to the mesenchymal stem cells because mesenchymal stem cells is kind of the very typical cell that have a great potentials in the skin disease. So the mesenchymal stem cell derived vesicles, those have a very good applications potential, in treat of the cardiovascular disorders, skin disorders, neurological disorders, and also can be benefit to the liver, lung, and the kidney injuries as well. So what happens to the axon zones released by the mesenchymal stem cells? Yeah, those small vesicles actually, they have a great potential and applications for skin rejuvenation because it can really accelerate the epithelium layers for the transitioning and remodeling, and it also can improve the extracellular matrices remodeling, okay, and the production. It can have the anti-inflammatory function. It also has the immunomodulatory function, and it also can promote the blood vessel growth. So this is the exosome and applications in the skin, mainly for the skin generation, regeneration, and rejuvenation. So I think those are probably the products going to be you may have it or in your clinical site, and potentially may be used for treatment of many types of the skin conditions. So those are the exosomes right now. Have been registered to the clinical trial.org, and then they have some information there. And then how their performance? Okay, today I'm going to look at this particular company, like the VT Labs in US. So they are produce those exosomes from the mesenchymal stem cells. They want to use those exosomes for treatment of the skin regeneration. Okay, so this is what happens. So today I'm here because I think it's better for me really to deliver the true and reliable information to everybody, not only to the physicians, also to the industry. Because as an academic person, I think everything needs to be truly based on the information and the fact. So this is what happens to the company. Okay, so the FDA basically issued a warning letter to the company, I think last year, okay, in August. Okay, what happens to this? Because they issued many of the violations, okay, so to the company. So see, you can tell. So this one, they said, oh, what you tried to use in the clinic is not consistent with what you said on the product labeling. Okay, so this is a violation. The second, I think what the FDA said, oh, your production basically is messy, something wrong. And then the third one, they said, oh, your product basically has no license yet, but you right now, distribute this to the clinical site. This is a huge, a very serious violation. And then the last one, I think FDA said, oh, your production is basically a violation of the CGMP rules, or you are doing these products in the CGMP facility. However, you do not follow the guidance. Do not follow the guidance of CGMP. And then if you look at specifically FDA's warning to what happens to these products, he said, okay, so first, failure to establish and follow appropriate written procedures designed to prevent. Okay, so those are about the contaminations. So it's about the CGMP, okay, procedure. The second one is also about the CGMP procedure. Okay, basically you do have a CGMP facility, but you do not follow. Okay, the third one is very serious. What he said is that failure to establish laboratory controls that include scientifically sound and appropriate specifications, standards, sampling plans, and test procedures designed to assure that components, drug product containers, closures, in-process materials, labeling, and drug products conform to appropriate and standard for identity, strength, quality, and purity. So basically, this violation is from the beginning to the end of your products. Okay, so and the third one is the failure to establish and follow written procedures for production and the process control. So then basically, you say, okay, you don't have control between the batch and the batch. And then you don't have control for the components, okay, and the materials. And then lastly, they said you don't have control for the product stability. Okay, if you are a biological product, what is the stability, okay? And what is your expiration date? You don't have any information about that. However, and then you push this into the clinical site. So this is a huge violation. Okay, and then this, I think this is the key message I want to deliver today, is that what is the scientific and the technical challenges, okay, for using stem cells and the exosomes that are going to be used for the skin rejuvenation and regeneration. So those, both of them are having great potentials. I believe in the next 5 or 10 years, those products are going to be in the clinic. However, we're still facing many of the barriers, and we need to overcome because they really can be used for the patient. So first, the SMC, meaning the mesenchymal stem cells. So first is that purification of these cells from different types of tissues. Okay, how are you going to purify it? Okay, so you need to purify it at least in the CGMP facility, and you need to have a good expertise. Okay, you need to have a very good cell biologist to handle it. Okay, and then how are you going to really define the differentiation directions of those SMCs to whatever you want? Okay, so for example, you want those skin to get more of the fibroblast, okay, to produce collagen fibers remodeling. And then you need to have the SMCs to differentiate into the fibroblast direction. Okay, if you want to promote the blood vessel regrowth, then you need to really promote the SMCs to the blood vessel side. Okay, so those are challenges right now. And the next is about the exosomes. Exosomes challenge is pretty huge. So first, whatever you got, the exosomes, those are heterogeneous population. Includes the exosomes, ectosomes, and also other macro molecules. Okay, based on current technology, it's very hard to distinguish exosomes from any other types of the vesicles. The second, the contents in exosomes are highly heterogeneous. Okay, it's very hard to control. This is the reality. Okay, this is the reality. The third is hard to determine which products or which components in your exosomes are effective. It's very hard, okay? Because your exosomes, what you got is actually, you may think this is a soup of the Chinese medicine. It includes everything, okay? So this is why it's also hard, for the quantity control, especially for the batch and the batch control. And it's very hard to get a consistency between different batches of the exosomes. So the last part, I would briefly talk about the regulatory compliance, because I think this is fundamentally important, not only for the physicians here, but also for industry. Okay, so the human substrate research is really important, and especially for the ethical guidelines. They have 2 very important documents. Those are the cornerstone documents. Okay, so one is the Helsinki, the declaration. Another one is the Belmont reports. Okay, so both documents basically set the guidance for the human research, ethics. And then every country has their own regulatory body. I think China is going to have the CFDA. Everybody knows. The CFDA is going to regulate or govern not only the industry, but also the healthcare workers, right? And also going to govern the relationship between the industry and the physicians. Okay, so, and then for the industry, they have to, have everything ready, preclinical setting, and then they can have these products, they can move to the clinical side and collaborate with physicians, and then try to generate a reliable and trustful clinical data. And they both, the healthcare workers and the industry partners, need to work together in a cooperation, in a collaboration, and then it also need to remain the independency. So this is the last slide I'm going to have. So this FDA actually is the boss, yes. They're going to ask you guys, physicians and the industry partners, okay, you need to comply, okay? And he's going to be very bossy, okay? And then, because better compliance with regulation and the better safety for patients. For the industry side, I think the QC is very important. QC on every step needs to be implemented. you need to be implemented. There is no gray zone. Okay? There is no shortcut. Okay? So, and then for the physicians here, you need to, every product, you need to ask your industry partner friends, license is ready? If not, I'm sorry, you get out the door, okay? You can come back when license is ready. The second, you need to make sure the scope of study is going to be aligned with what the product's labeled. Then you need to make sure your IRB is ready, okay? And when everything is ready, yes, you can go, because that's the guarantee and the insurance for the human subject research, ethics. Okay, so I think I'm going to stop here. Thank you so much.